Antibodies, also known as immunoglobulins, or Ig for short, are crucial components of the immune system. They are glycoproteins produced primarily by B cells, which are a type of white blood cell, in response to the presence of antigens. Antibodies play a key role in identifying and neutralizing foreign substances like bacteria, viruses, and toxins. Structure of antibodies. Antibodies are Y-shaped molecules composed of 4 protein chains, 2 identical heavy chains and 2 identical light chains. These chains are held together by disulfide bonds and form 2 important regions: the Fab region, or fragment antigen binding, which is the variable region where antibodies bind specifically to antigens, and the Fc region, or fragment crystallizable, which is the constant region that determines the antibody's class and mediates interaction with immune cells. Each antibody has 2 antigen binding sites, making them bivalent, allowing them to bind to 2 identical antigens simultaneously. Types of antibodies. There are 5 major classes of antibodies, each with a different role in immune defense. First, IgG, the most abundant antibody in blood and extracellular fluid. IgG provides long-term immunity after infection or vaccination and can cross the placenta to protect the fetus. Next, IgA is found in mucosal areas, such as the gut, respiratory tract, and urogenital tract. It is present in secretions like saliva, tears, and breast milk, protecting mucosal surfaces. Then IgM is the first antibody produced in response to an infection. It is efficient in forming antigen-antibody complexes and activating the complement system. IgE is involved in allergic reactions and defending against parasitic infections. It binds to mast cells and basophils, triggering the release of histamine and other chemicals. Finally, IgD is present in low levels in the blood and functions as a receptor on B cells during early immune response stages. Antibodies protect the body through several mechanisms. First, there's neutralization. Antibodies bind directly to pathogens like viruses or toxins, preventing them from attaching to and entering host cells. Then there's opsonization. Antibodies mark pathogens for destruction. When bound to an antigen, the Fc region of the antibody attracts phagocytes, such as macrophages, which engulf and destroy the pathogen. Another mechanism is the activation of the complement system. Some antibodies, especially IgM and IgG, activate the complement system, a series of proteins that assist in lysing or breaking down pathogens. Additionally, antibodies can bind to multiple antigens at once, causing clumping or agglutination. This makes it easier for phagocytes to engulf multiple pathogens simultaneously. And finally, antibody-dependent cellular cytotoxicity, or ADCC, antibodies bind to antigens on infected cells, marking them for destruction by immune cells, such as natural killer, or NK cells. Antibody production begins when B cells are activated by an antigen. This activation triggers a process called clonal expansion, where B cells proliferate and differentiate into plasma cells that secrete large amounts of antibodies, and memory B cells that provide long-term immunity by remembering the specific antigen for future responses. Antibody diversity is achieved through gene rearrangement. The genes encoding the variable regions of antibodies undergo rearrangement to create millions of different combinations. Additionally, somatic hypermutation occurs. Once B cells are activated, their antibody genes can mutate slightly, producing antibodies with an even higher affinity for the antigen. Vaccines stimulate the immune system to produce antibodies against a specific pathogen, providing immunity without causing the disease. Monoclonal antibodies. These are laboratory-made antibodies designed to target specific antigens. They are used in treatments for cancer, autoimmune diseases, and infectious diseases. Autoimmunity. In autoimmune diseases, the body's immune system mistakenly produces antibodies against its own tissues, leading to inflammation and tissue damage, allergies. In allergic reactions, IgE antibodies bind to harmless substances like pollen, causing an exaggerated immune response. Antibodies are essential for defending the body against infections and play a critical role in various medical applications. Understanding how antibodies work not only enhances our knowledge of the immune system, but also aids in the development of treatments for diseases, including immunotherapies and vaccines.